The natural world is filled with familiar shapes and patterns that appear time and time again. This order and regularity is a feature common to both inorganic and living things. The shapes, colors, textures that we see in plants and animals are not just simple products of natural forces. These patterns are passed on from one generation to the next, a reflection of their biological inheritance. The wings of butterflies display a myriad of color patterns and provide us with one of the most striking studies of inheritance. Each butterfly inherits information from its parents in its genes. But how is this information translated into color patterns? Does the environment have any influence? And by what process are patterns formed during development? To answer these questions, we need to examine the role of genes in the context of changes that take place during the critical stages of a butterfly's life cycle. In this South American butterfly, the genes involved in pattern formation have been identified by breeding experiments. Species of Heliconius are a collector's delight, but a classifier's nightmare. There are a bewildering array of color patterns, but they've been shown to follow simple rules of inheritance, as John Turner explains.
There are only about 40 or 50 species of Heliconius in South America, but the group has produced somewhere between 600 and 800 different named varieties that collectors have given fancy Latin names to. And any one species can produce up to 50 recognizably different patterns. At the same time, two butterflies that look exactly the same can actually belong to two completely different species. So that the group is really very difficult to sort out. And it's also quite obviously undergoing some very interesting explosive evolution in its color patterns. So that these butterflies are extremely interesting to an evolutionist. Now, if we want to find out about the color patterns, what we need to do is to breed the butterflies. All you need is a warm greenhouse and the passion flowers, which the caterpillars feed on, and various bits of food for the butterflies themselves. Now, most butterflies, if you try and breed them in a small cage to get the offspring of single females, which is what you need to do if you're doing genetics, the butterfly simply wears its wings out on the fabric of the cage. Now, that's where you win with Heliconius, because a single butterfly knows its territory. They learn the boundaries of the cage, and they come up to the mesh and fly away again. They don't batter themselves to death. So they're actually one of the few butterflies that you can keep in a comparatively small cage in captivity. Carefully, that's it. Right. What we do is we take two butterflies with very different patterns from different parts of South America that actually belong to the same species and mate them together to see how the patterns are produced.
Okay. Right. That's lovely.
Two we need.
Okay. Excellent.
We've got the results of an experiment like that here. We've crossed a butterfly from the mouth of the Amazon with one from southern Brazil. Now, you can see those butterflies have got very, very different patterns, but actually they belong to the same species, and they'll produce fertile offspring. What are the offspring going to look like? Well, they all look like this, and it's sort of intermediate between the two parents, but we can notice certain things about it. One is that this big yellow bar here across the wings has disappeared in the hybrid offspring. We say in that case that the yellow bar across the wing is recessive, and that the absence of the yellow bar, is dominant. And the same thing happens with this great sunburst pattern on the Amazon parent. That's dominant, and it appears in the offspring, and the absence of it, which comes from the East Brazil parent, is the recessive character. So the dominant characters appear in the offspring, and the recessive ones don't. So what's happened to the recessive characters? Have they just disappeared? No, they haven't. The fundamental discovery that Mendel made 100 years ago when he started breeding experiments with peas was that recessive characters do not disappear. The genes which produce them remain perfectly intact and still exist in that hybrid butterfly. The gene that produces the yellow bar is carried by that butterfly, it just isn't expressed. And we can show that that butterfly carries it by doing another bit of the breeding experiment. What we do is to take one of the sisters of this individual and mate them together. And if we do that, out will come another generation of hybrids, which we call the F2 generation of hybrids. That's the second generation of hybrids. And we've got those here. Now, you can see what happens there is that all the variation is let loose. And that looks very complicated, but it's actually quite simple when you know how to interpret it. Let's just look at that yellow bar. The yellow bar has reappeared in the second generation of hybrid butterflies. What's more, it's appeared in a regular way. About 1/4 of the butterflies have the yellow bar, and 3/4 of them do not. And that's characteristic of recessive characters, that they come out in the next generation in a ratio of 1 recessive to 3 dominant. And if we look at what happened to the sunburst, you can see exactly the same thing happening. That's appearing, because it's dominant, in a ratio of 3 with the sunburst to every 1 butterfly without the sunburst. Now, one other thing we can notice about this is that the sunburst and the yellow bar are independent of one another. We say that the genes are inherited independently. We have all 4 possible combinations: yellow bars, no sunburst; yellow bars with a sunburst; no sunburst and no yellow bars; and just the sunburst by itself. So what we've got here is genes producing the patterns of butterflies. These are patterns that are inherited.
But just how do genes produce patterns? And when do genes exert their influence? One clue to the process is suggested by the similarity between the eye spots we see on butterfly wings and certain patterns produced by simple chemical reactions. Waves of coloured rings are formed in a reaction named after its discoverers, Belousov and Zhabotinsky. And these concentric circles, known as Lisagong rings, are precipitated by the interaction of 2 chemicals. The fact that they resemble eye spots suggests that the underlying mechanism in butterfly wings might also involve some basic chemical process. Dr. Fred Nijhout.
Now, the first problem in analyzing pattern development in this particular system is the fact that pattern itself gives you very little information about the process that gives rise to it. The circular patterns, for instance, in the Belousov-Zhabotinsky reaction and the Lisagong rings, while they look very much alike, come about through very different chemical mechanisms. So if in butterfly wings we would wish to find out how eye spot patterns arise, we are going to have to examine this experimentally by somehow interfering with the process that we believe gives rise to these eye spots. Now, we've been doing most of our experiments on a small American butterfly, Precissinia, the buckeye butterfly. And the really nice thing about this species is that on the cuticle of the pupil wing, there are a number of small marks that exactly mark the center of the eye spot that will develop on the adult wing that lies underneath. So that what we can do then is to experimentally go in and attempt to interfere with that small group of cells that lies exactly at the center of the eye spot. We do this by a procedure known as cautery. We very simply go in and touch this group of cells with a small wire that has been heated electrically. We kill those cells by doing this. And then we allow the animal to continue to develop. And in the adult, when it finally emerges, we see whether this surgery has somehow affected or interfered with the color pattern that develops. Let me show you the results of an experiment in which we actually removed and killed the cells at the center of an eye spot. Here's a specimen, for example, in which the cells at the middle of the eye spot on the left-hand wing were killed, and you can see that the development of the eye spot was completely inhibited in this specimen. Now, we're only killing a very small group of cells, and when we do this experiment over different times after pupation, we find that about a period of 48 hours is required for eye spot determination, and that this period occurs about 5 to 7 days before the pigments will actually develop on the wing of the butterfly. Now, the number of cells that we kill in one of these experiments is about 200 to 300, which is a very small number. It is about maybe 1% of the total diameter that the eye spot will finally grow to. Now, to demonstrate that these cells at the center of the eye spot indeed are very important in eye spot determination, we can, instead of killing them, also simply cut them out and transplant them to a different location of the wing where normally no eye spot would develop. We remove a small area of cuticle from the animal that we are going to use as a host, and into this we transplant a similar sized small area of cuticle from the donor, and the animal is then placed in an incubator for development. After a week, the adult comes out, and we can observe on the adult wing the effects of the transplant of the experiment. Now, what is really nice about butterfly wings is the fact that underneath the cuticle is a single layer of cells called the epidermis that contain all the cells that will make up the surface of the wing. In this epidermis, there's no cell movement, there's no growth, and so what we are observing is really pattern formation in its purest sense, undistorted by the movement of cells. Here we have the results of an experiment in which we transplanted a focus from one specimen onto this area of the wing of a different specimen, and you can see that around the focus in the host tissue there's developed a small miniature eye spot. Evidently, the cells of the transplanted focus have emitted some signal that has caused the cells of the host and the surrounding to manufacture the pigments characteristic of an eye spot. We have reason to believe that the signal that is emitted from the cells of a focus is a small diffusible molecule. Presumably, this molecule has an effect on the cells that it diffuses to, in that it causes these cells to activate or inactivate the right kinds of genes that are involved in the manufacture of particular pigments.
But how could such a variety of shapes and colours be determined by a diffusible molecule? Imagine a chemical signal is produced at the centre of a group of cells. The highest concentration will always be at the source, decreasing as the signal diffuses away from it. A cone-shaped concentration gradient is set up, with a circular pattern developing out from a point source. If cells respond to decreasing concentrations of the chemical by producing less pigment, then the result will be an eye spot with concentric rings of colour. More complicated patterns could be produced if, for instance, in addition to a point source, there is a line source at the edge of the wing. By variations in the positions and types of source on the wing, it's possible to imagine how a great variety of pigment patterns could be generated. The species Heliconius melpomene has as many as 12 races, each with a different pattern depending on where it's found in South America. For example, it has this pattern in Venezuela, this one in Ecuador, looks like this in the Amazon basin, and like this in southern parts of Brazil. Breeding studies show that only a small number of genetic changes are needed to produce these diverse patterns. Once we know which genes exist in present-day races, we can work back to find out what the ancestors may have looked like. Reconstructing an evolutionary tree is an exercise that involves logic and a careful application of genetics, as John Turner explains.
We can do this by playing around with the patterns in coloured paper. Now, I'll show you how it's done. Let's suppose we have two races, one of which has the yellow bar and the other doesn't have the yellow bar. Did the ancestor have the yellow bar, or did it not? Now, one of the things we understand about evolution is that natural selection finds it much easier to replace a recessive pattern with a dominant pattern than it does to make evolution happen the other way around. So we can use that information to decide which of these two possible patterns was the ancestor. Let's start off by supposing that it was the one without the yellow bar. Now, in that case, something rather difficult happened. Natural selection caused a recessive pattern to appear in the population. That's difficult. It probably didn't happen that way. On the other hand, if it was the yellow-barred pattern that was the ancestor, then we have a much easier evolutionary tree. In this branch of the evolutionary tree, a dominant pattern appeared in the population. In this branch of the evolutionary tree, there was no change. So the most probable solution to this question is that the ancestor of these two butterflies several thousand years ago looked like that. Now, we can go on doing that with all the present-day races, and we can reconstruct the whole of the evolutionary tree back tens of thousands of years into the past.
To trace back the Heliconius melpomene family tree, John Turner started with present-day races. He chose the ones whose genes are best known and paired those that are most alike genetically. This is the pair that he traced to an ancestor with a yellow bar. For this trio, it's the black rear wing that's recessive, whereas the red rays are dominant. So the ancestor's rear wings would have been black and without rays. Sometimes the closest relative of a modern race may be an extinct ancestor. These can be joined to an even more remote ancestor. The remaining two races must have descended directly from the great-grand ancestor of the whole species. Its pattern, he thinks, consisted entirely of the recessive characters, a black butterfly with yellow bars across its wings. It probably existed sometime before the last ice age, over 30,000 years ago. Only through a knowledge of the genes of living butterflies was he able to resurrect the ancestral relationships. But why do butterflies evolve such different colour patterns? In Heliconius, part of the answer has to do with protection.
One of the things the patterns of these butterflies have to do is to protect them against predators. The bright patterns serve notice to birds and lizards that these butterflies are nasty. They're full of cyanide and probably various other things. And the bright warning patterns remind the lizards of that fact, and the birds. Not only do they advertise their nastiness, but they're in advertising syndicates as well. Heliconius melpomene has a running mate, Heliconius erato, which flies with it everywhere in South America and looks just like it wherever Heliconius melpomene goes. They're different species, they've got different caterpillars, they feed on different vines, and what's more, if you sniff them, Heliconius erato smells of witch hazel, and Heliconius melpomene smells of fried rice.
Although these two caterpillars look fairly similar, the one with the black stripe down the side of its body is erato, and melpomene has an orange head. In the pupa, erato has the longer horns. And in the adult butterflies, erato can be distinguished from melpomene by having more red dots on its thorax. But otherwise, the two species are almost perfect mimics of each other. It's a well-established double act. and pairs of mimics turn up wherever these species are found in South America. When John Turner compared his Melpomene tree with the one he'd constructed for Erato, he was surprised how faithfully they'd maintained their mimicry. There have been only slight differences between the species throughout their evolution.
There isn't one explanation for a butterfly's pattern. There are four explanations. First, there's development, the way in which the pattern is spun out on the wing by the genes and the environment. Second, there's a genetic explanation, what genes the butterfly received from its parents to cause the development of the pattern to be triggered in that particular way. Third, there's the function of the pattern, what this pattern does for the butterfly, what natural selection has given the species in the past, in this case, the protection of the butterfly from its predators. And finally, there's history, what patterns the ancestors of this butterfly had over the past many thousands of years. With a different history, it would be a different butterfly.
Although changes may take place over the course of evolution, during the lifespan of a heliconius butterfly, its pattern is stable and resistant to change. The colour patterns of other butterflies we've seen, like Pressis senior, are more susceptible to manipulation. By exposure to temperature shock during development, it's possible to induce severe changes in pattern. Butterflies whose patterns are altered by an environmental change can sometimes resemble another species altogether, with a totally different genetic makeup. These are called phenocopies.
I have here in this drawer, for instance, some phenocopies that we induced by temperature shock in Pressis senior, and these specimens here look very much like the normal pattern of a closely related species that occurs in the southwestern United States. Now, the important part, or the important feature of all of this, is that form or pattern as we see it is the endpoint of a developmental process, and that this developmental process comes about through an interaction of both genes and environment. Those of us who are interested in studying this interaction can study this at two very different timescales. Those of us who do developmental biology tend to study the interaction of genes and environment over a very short timescale, usually the lifespan of a single individual, whereas those of us who are more interested in evolution study the same kinds of interactions, but over a much longer time scale, usually spanning many generations. Ultimately, however, if we are to discover how genes control form, it is necessary to study the interaction of genes and environment at both of these time scales. Only in that way will we ever be able to draw the connection between gene and form, and also to discover how and why genes and form change over time.